Hello guys, welcome to Learning with Isha. In today's video, we're going to talk about a technique known as tissue culture. So what is tissue culture? As the name suggests, tissue and culture. Naturally, it involves something related to tissue and the culturing of the tissue, means growing of the tissue. So why are we growing this tissue? First of all, we are growing this tissue, a plant tissue, to get an entire plant. This technique was started by scientists in the early 1950s. What is tissue culture? Tissue culture is the regeneration of an entire plant from a small part of the parent plant. So this means that we are going to take a small part of the parent plant and then we are going to grow it in some way to get a new plant. In tissue culture, we'll understand how we are growing it. Now, what is the part, this old part of the plant called that we are growing, right? It is called explant. It is called explant. X means what? Old. Okay, so it's called explant. What is explant? The part of old plant cultured to get new plant. Okay. So what parts of the old plant can be cultured, right? Practically any part of the old plant can be taken. Okay, we can take seeds, we can take embryo, we can take anther, we can take pollen, we can take leaves, a part of the stem, anything can be taken. One cell from this is obtained, or a small tissue is obtained, it is grown and we get a new plant. Now for tissue culture, we require two things, okay? We require two things. What are those two things? First, we need a sterile environment. What do we mean by a sterile environment? See guys, we're taking one cell, we're trying to grow that cell, right? So I wouldn't want my new plant to be infected with some kind of a pathogen, having some kind of a disease. So to maintain a disease-free condition, to maintain a bacterial or viral infection-free state, I would require a sterile environment, means a contamination-free environment. The second thing that I require to grow my one cell is a nutrient media. A nutrient media. What is a nutrient media? Something that has nutrients naturally. Why do I need nutrients? See, I take up food, that food is digested in my body, I get energy out of it, right? It helps my body to grow, to form new cells for the maintenance of my body functions. Similarly, this one cell has to grow into a new plant. So it needs energy, right? So it requires nutrients. What are those nutrients? First, I need a carbon source. I need a carbon source. Second, I need a source of nitrogen. I need a nitrogen source. Let's understand these two first. Carbon and nitrogen are called the structural elements of cell, right? If I have to make a protein, I need carbon. If I need to make a nucleotide, I need carbon. For enzymes, for other things in the cell, I need carbon and nitrogen, right? Where can I get my carbon from? Okay, I can get my carbon from sugars like sucrose. It is a sugar, it can give me carbon. What compounds in the cell are rich in nitrogen? Proteins are rich in nitrogen. So for nitrogen, I'm going to add amino acids. Okay, proteins have nitrogen. So I'm going to add amino acids, so I'll get a nitrogen source. Okay, besides that, I require vitamins, minerals. These are growth supplements, okay? Also, I am going to add certain growth hormones. you must be knowing about plant growth hormones, right? Auxins, cytokinins, gibberellins, right? So here I'm going to add auxins as well as cytokinins. Okay, this is going to help my cell form roots and shoots. Okay, so this is the nutrient media that I require for tissue culture. Okay, now tissue culture is also called as micropropagation. It is also called as micropropagation. Why is tissue culture called micropropagation? See, micro means small. Propagation means forming new, something new, right? Propagating. So micropropagation tissue culture is called because new plants are formed from a very small part of the old plant. Okay, new plants are obtained, and many new plants, in fact, obtained from small part of the old plant. Small part of the old plant. That is why it is called micropropagation. I need a very small amount of tissue, from that I can grow many plants, right? Now also in tissue culture, the new plants that are obtained, we have a term for that as well. They are called somaclones. Somaclones. Somatic cells of the body. What are somatic cells? The normal cells of the body are called somatic cells, right? So somaclones. They are called somaclone means identical copy. So they are called, the new plants are somaclones. Why? Because they are identical to parent plant. They are identical to parent plant. That is why they are called somaclones. Okay. Another important terminology that is used in tissue culture is called totipotency. Okay. What is totipotency? See, one cell is going to divide in the presence of a nutrient media in sterile conditions into one ball of cells. Okay, this cell is going to divide and form a ball of cells. Okay, and this ball of cells is eventually going to form my new plant. So now this ability of one cell to divide into a ball of cells and form the new plant is called totipotency. Totipotency. As one cell from any part of the whole plant, stigma, style, anther, leaf, stem, irrespective of where it is coming from, it is making this ball of cells and forming a new plant, right? What will this ball of cells be called? This ball of cells will be called callus. This ball of cells will be called callus. So one cell forms a callus, callus grows and forms a new plant. Okay, now here in the callus, this cell might go and make the shoot. Let's say this one makes the root. So there is no any differentiation right now what part, which cell will form which part of the new plant, right? So such a group of cells is called undifferentiated cells. Undifferentiated cells. Why? Because I don't know which cell is going to form which part of the new plant, right? Undifferentiated cells undergo what? They differentiate and form new plant. Okay? यहाँ तक समझ में आया? Fine. So now we are going to move on and see where it is being commercially used. Tissue culture is commercially being used to form new plants in the case of tomato, banana. Okay, plants like this, it is being used to form a new plant. Apple is another example. So these are the three examples given in NCERT, and one must remember them, right? There might be a question, you'd be given some other option as well. You might be given lemon, right? You will be asked which one is being presently used commercially to form a new plant. So it is tomato, banana and apple, okay? They are being used. Fine. So now that we know a little bit about tissue culture, let's move on and understand the mechanism of tissue culture, right? Now let's move on and understand the mechanism, okay? What is the mechanism? What are the steps? Right? The mechanism of tissue culture. Firstly, anything starts with an initiation, right? I initiate something. What does it mean? I'm starting something, right? It is called initiation. So the first will be what? An initiation phase. Okay. What is going to happen in an initiation phase, right? I'm going to take one cell. Okay, from what I told you, any part of the whole plant, I'm taking one cell. I'm going to add it in a nutrient media, give it sterile conditions. This is called what? Initiation, right? I'm giving conditions that are appropriate for this one cell to start dividing, right? Now, naturally, when my cell gets these appropriate conditions, what will it do? It will start dividing. It will start multiplying. So there will be a multiplication phase. Okay, there will be a multiplication phase. What is multiplying? My one cell is multiplying to make many cells. I told you these many cells, this ball of cells will be formed called callus. So this is happening in my multiplication phase. Now that I have callus, what did I tell you? There will be differentiation now, right? This callus is now going to differentiate. It will differentiate from specific parts of the new plant. So what happens is a root starts to form, okay? There is a root formation phase after this. Now my callus will differentiate. My callus will undergo differentiation, okay? And a small root-like structure will start to appear, okay? Followed by what? Root ban gaya, shoot formation, okay? So there will be shoot formation. What is root and shoot? I hope you know, right? See, this is a plant. What is below the soil is root. What is above the soil is called the shoot, okay? Just for your knowledge. Anyways, moving on. Next phase is now I have roots. Now I have a shoot that has started to form. Now what will I do is, now I have small, what we call as plantlets, okay? The very small plants, they are called plantlets. So now these plantlets are growing in lab conditions. When we grow something in lab conditions, what is it called? In vitro culture. It is called in vitro culture. Okay? In vitro means growing something outside the body. Okay? It's called in vitro. So what does growing something inside the body, what will it be called? It will be called in vivo. Okay? This is not in vivo. I'm writing it here. Don't confuse. It is not in vivo. This is in vitro. This is happening where? In a lab, in a test tube. Okay? So this is in vitro. Now my plantlets have formed. Now they are in the lab. Okay, fine. Now I want them to adjust to the natural environment. I'd want them, to grow in normal fields. I'll not want them to grow in the lab, right? So what I'm going to do is, I'm going to acclimatize. Acclimatization phase. What is acclimatization, guys? See, if you decide to go on a vacation to Leh Ladakh, okay, fine. What is going to happen? The government is going to set some rules for you, right? It's going to say you cannot straight away go there and start enjoying your vacation. You need to get adjusted to the temperature, right? In Leh and Ladakh, the temperatures are sub-zero. It's very cold, okay? Also, the pressure in the air is low because of the high altitude, right? Oxygen levels are low. My body will need some time to get adjusted to the change in environment. Let's say I'm staying in northern part of India where the temperature doesn't go that cold normally, and I suddenly decide to visit Leh and Ladakh, okay? My blood cells are not ready to transport oxygen in such a low atmospheric pressure, right? I need more RBCs in my body. So for that, I need to give some time to my body, right? That is called acclimatization. So now these lab plants, before I get them into the real fields, I need to give them some time to adjust, right? So first what I'm going to do is, in the first phase, I'm going to grow them in greenhouses, okay? Greenhouses, and after that I'm going to grow them in the nurseries. Okay, now my plant has come to the normal fields. So in short, let's recap what we did. I took one cell from an old plant. I wanted to get a new plant. I gave it appropriate conditions and I got a new plant by a technique known as tissue culture, also known as micropropagation. Okay, the new plants that will be formed here will be called somaclones because they are identical to the old plant. Okay, now in the next step, what we are going to see is the advantages of tissue culture, right? I could have simply done the traditional hybridization techniques, right? The traditional farming. I could have put the seed in the field and just done that. Why do I need to do this technique, right? So that we'll see in the next video. Thank you.